So, okay. All right, we're still getting used to this new platform. So for speakers, when you present, you should click on the webinar session. So Filippo, you need to click on the webinar button on the bottom. But if you want to do something else or mute yourself, that's totally fine. And you can also click on the backstage at the bottom of this bar, and then you will be invisible from the event. And you can still watch the event. You just have to adjust the setting of the audio. So let me just force Filippo to go to the webinar. Here we go. I'm just gonna force everybody to go to webinar. Let's see if this is working. There you go. Now you're in the webinar. So we're using this new platform where you have a backstage and you have a webinar kind of thing to simulate real stage. So welcome everybody. We have slight technical challenge this morning, but and I messed up a little bit this morning, but still we are on time, all here, and really excited to present this panel. My name is Chen. I'm the founder and CEO of 3D Heals. We've been here for years and years with three main missions. One is to educate everybody about all the ongoing cutting edge technologies in 3D printing and 3D technologies. As you can see, we include both bioprinting and also AR, VR as well in this category of 3D technologies. And number two is we're trying to create an environment where people can network and collaborate and found companies or projects. So that's number two. We have pivoted to virtual because of the pandemic, but we're definitely working towards a hybrid format in the near future. And number three is I help early stage startup companies like Filippo's company. Actually, he will tell us very soon with fundraising. It's very tough to get money when you are a 2-people company or 1-person company. And I'm here to help. We have a program called Pitch 3D. It's free. We're not getting paid. Nobody's paying us for anything. We're just a facilitator to connect startups with VCs and institutional investors and also large angel groups. So I think today's topic is pretty relevant. It's very timely because there are so many hot topics out there talking about OpenAI, Microsoft, Google competition in this platform. But within our small industry, 3D printing and 3D technologies, I think it's very relevant to talk about how this new momentum can help pushing the industry forward. And we're already seeing a lot of innovations here. And everyone who are speaking today are already years deep into, basically working in this space, connecting AI, machine learning, and 3D technologies. So I'm pretty excited just to learn what the speakers have to say today. I also want to have a shout out to our sponsor today, Synopsys Simpleware. Karim is actually our speaker from the sponsor side, but their product is actually quite amazing. So, but I'm not going to talk more because he obviously knows more about the product than me. So I would like to introduce our first speaker, and it's Dr. Philippos Tolomosis. I am hoping I did an okay job with that name, but I met Philippos because his social media posts and the works that he published, and I was just really fascinated with it. I've seen a couple of papers published on AI, machine learning, and bioprinting, and I thought he actually is commercializing his work involving the area, not entirely, and I just find it very fascinating and exciting. But he also has a lot of academic positions as well, a very extensive international background in scientific communities. And Philippos, I'm not going to occupy the stage too much. I'll let you take over. You can introduce yourself as well. And I look forward to your presentation.
Yeah, thank you very much. Can you see my screen?
Yeah.
Perfect. Yeah, thank you very much. Really excited to be here and present again. And thank you very much, Jenny, for all the help in these early stages. So I'm the founder and the CEO of BLI, which is a startup manufacturer of architected biomaterial scaffolds, as you can see in the background of this image. And those scaffolds have geometrical feature sizes down to the single cell level size. And we are commercializing those scaffolds in order to empower scientists to study and master human biology in 3 dimensions in a very unique way, as I'm going to explain in the following slides. A couple of words about BLI. It's a US company, venture-backed. 1 year ago, we started R&D operations in Athens, in Greece, where is my home country. And the reason about that is because we got a large infrastructure research grant that is helped us support, 6 people working in the R&D section of the company, and we have managed to raise some pre-seed money in order to fulfill our mission. And our mission is to empower scientists to study cells in 3 dimensions using scaffolds, but not using them only as supporting structures, using those geometrical feature sizes that you are able to design and manufacture as programs in order to guide cell function. And I'm going to step back and, we know that when we do biology, we start with cells in a vial. They have a circular shape, and the goal is always to plate them in some sort of a 2-dimensional surface, along with all the media and the chemical molecules and the food for the cells. And the goal here is those cells, they start to expand, they change shape, and we want to expand them in number and use them and harness them from that 2-dimensional surface for many different types of studies. And the goal is to guide always the cell function for our application. But there is a problem, and the problem is that we always know what is the phenotype or the function in the beginning, but in biology, as it has been documented in many papers recently, there is always this reproducibility issue and this randomness. And in order to achieve this cell function goal, we use chemical signals about that. So our solution is our 3-dimensional scaffolds, which is not a new idea, it's an old idea. But the new idea here is that you use the scaffolds, as you can see on the left picture where you see humans climbing a steel scaffold. Those humans are experiencing different stresses as they adopt different cell shapes. So if you are able to have a similar structure down to the single cell level, you are able to change and tune cell shape. And as we know, form guides function, and you can guide cell function with amazing resolution down to the single cell level, opening a whole new design space of opportunities. Basically, what we are doing is we are taking the classical dogma of biology, which is, as we know, we go to the DNA and we do some gene editing, and then the transcription phase is happening, and the goal is at the translation phase to change those proteins and guide them in order to guide cell function. But what if you could use architected biomaterials and scaffolds to do exactly the same way by reversing it? And I think this is a very profound idea because you can do it very reproducibly. If actually the scaffold that you are designing is inducing DNA changes down to the single cell level, this scaffold is being designed and manufactured by a machine. So you have a very engineered and reproducible way to do biology. So we are filling this dimensional scale window of synthetic polymers. As you can see, there are products on the left side. We all know about nanofiber membranes that are being manufactured with electrospinning technologies. On the right side, you can see classical architected biomaterial scaffolds that can be manufactured with FDM 3D printing or extrusion-based 3D printing technologies. And we come in this middle, is missing dimensional scale window, and we offer those scaffolds that give us the opportunity to be down to the single cell level, but also with absolute precision in the porous microarchitecture. Because the topic of today's webinar is machine learning, much of this research, in order to show that those scaffolds can be used to manipulate single cell shapes, but also to show that it can be done in a very homogeneous way, okay, in order to assess the non-reproducibility issue and the heterogeneity issues in biology, we employed machine learning tasks. And what we did basically is we were printing scaffolds, we were seeding them with biological human cells, basically mesenchymal stem cells that you harness from the bone marrow. And we showed in this publication that, in a quantitative way, using a classification task, that indeed with those scaffolds, we can induce in a very homogeneous way specific cell shapes and guide cell function. We are selling right now our scaffolds to our early innovators network in 2 different architectures that you can use for stem cell expansion differentiation. Please reach out to me personally if you want. And we are launching a larger scaffold architecture product line because, in fact, with what I'm going to show about the platform and how we make those scaffolds, we have a design space of 30 million fabricatable architectures. And also, a bit later, we're going to launch also a foundry service composed of different types of, products, like machine as a service product. If someone wants to print his own scaffold in his lab, in his space, and a software as a service product, it's a design tool because, there are many design tools out there and amazing programs, but the process here has some intricacies that the current design programs cannot fulfill. The way we design those scaffolds is obviously with computational geometry, but there is a big constraint. The process is continuous. So when you start, you cannot stop. So in this way, you need to have intelligent, paths for the machine in order to achieve, different architectures. And then from those architectures, you want to tune your cell shapes to specific cell functions. So the first module is the design tool, and you want to couple it with some sort of a simulation because always when you want to design scaffolds, you want to map the mechanical properties. And if you are designing a medical device, an implantable medical device, human tissue has very complicated viscoelastic anisotropic mechanical properties. So we are trying to put that in a loop, okay, and search over all the possible designs, in order to find architectures based on specific optimization goals. And that is match scaffold mechanical properties with specific human tissue mechanical properties. And the way we are doing it, we are using, probabilistic machine learning. So every time we run a simulation, we map in a data-driven way a model, a surrogate model that gives us also the uncertainty. And based on that, we do active learning using the uncertainty as a handle. It's called Bayesian optimization, and people who are familiar with machine learning, they will know about this. After that, you send your scaffold to the machine, and we use machine learning also for the automation of the process. We want to have online quality control, so we are using computer vision, but not deep neural networks. We classical image processing techniques, very optimized in a multi-threaded way to do it in real time, and also extracting features during the printing process, both in the initial stages to calibrate the machine and stabilize it, so you don't have any reproducibility issues per printing job or per printing machine. We recently published our framework at this journal here at Nature Communications Engineering, talking about how we do that. Our goal, our foundry, is to have everything together and couple it. So this is our team. We are a team of bioengineers, mechanical engineers, and software engineers that love to do biology by looking at shell shapes. And if you want to reach out, please free, and we can discuss more and see how we could partner, collaborate, or whatever else. Thank you very much.
Thank you, Philippos. Hey guys, if you have any questions, feel free to put in the QA box. I have a few questions just to get some updates from you since we last spoke. What are some of the major applications that you're currently focusing on using your technology?
Yeah, the major application that we're focusing on is stem cell differentiation. So using mesenchymal stromal cells that have, a trilineage, they can become 3 different types of cells. We are trying to find ways to guide them at specific differentiation pathways. But also, and this is very, very important because we have clients that are interested how they can harness the differentiated cells, either for bioprinting purposes downstream or for organoid development. And then the other thing is trying to find architectures. We have a patent that the USPTO granted for these two architectures where actually you don't differentiate the cells, but you replicate them by maintaining their stemness, which is great for cell therapy applications. You want to deliver them in a very homogeneous way and keeping their stemness for the therapeutic potential. So yeah, it's stem cells, basically.
Awesome. And then these stem cells are going to differentiate into different lines of cells, I assume. And what are some of the major, areas of, end organ application that you're looking at?
So right now, we offer this tool for other people who want to use them for their own applications. If they can find it, I cannot, disclose because we have clients who are using scaffolds for that. But we are interested a lot in adipose tissue engineering for breast tissue reconstruction. We have a long-term plan for, next generation resorbable implants for breast tissue reconstruction.
Okay, cool. And then, so this tool is specifically using electrowriting or melt electrowriting. And
combined with FDM.
Yes. Oh, and combined with FDM. And does both have the machine learning, machine vision component to it?
Yeah, we plan to span out the machine as a service, product, where you have the machine and you have multiple melt electrowriting heads if you want to increase your throughput of your scaffold fabrication for melt electrowriting, or you have melt electrowriting plus FDM extrusion, and the system always has computer vision and all the other perks together.
And which one is harder to study between FDM and melt electrowriting?
Yeah, so it's, melt electrowriting because the process has, in the beginning, but this is solved now as a product because we are going to offer it within the software, the process, because it uses electrical fields, you see those instabilities and you need to be able to capture them and closing the loop by tuning the process parameters to maintain a stable jet. So yeah, melt electrowriting, I would say. Yeah.
Awesome. Well, thank you, Philippos. I'm sure we'll have some other questions later on, but thank you very much for an awesome and quick presentation. And I'd like to introduce our next speaker, who is Karim Jenk. He is the product manager for Simpleware at Synopsys. Karim.
Hi, Jenny. Can you hear me?
Yes, perfect.
Perfect. All right, and let me go through the process of sharing my screen here. Okay. All right, can you see my title slide now?
Yep.
Perfect. Okay. So yeah, thank you, Jenny. My name is Karim Gench. I am I'm product manager for the Simpleware group within Synopsys. And today I wanted to thank everybody for attending and also talk about how we leverage AI tools and machine learning to convert 3D images into models for patient-specific workflows, and how we leverage the technology to automate what tends to be a pretty manual process. So just to give you a little bit of background on the Simpleware product group. So we are developers of a high-end 3D image processing software and solutions. So from the high level, it's 3D images to models. That's what we do, and that's what we focus on. We have a dedicated sales and support and services team that have a global presence, and we work in primarily clinical and life sciences, but also we have other materials and manufacturing types of applications. But all in all, we are a part of a large software company, and we license our software for people to convert 3D images into models. So the 3D image-based modeling workflow is pretty straightforward. I think most people here are, I'm sure, are quite aware of it and how it works. But just to give you a bit of a background, Simpleware is what you see here in this gray box, where you have the image processing, where you can import your DICOM data, your CT or MRI scans of patients, and go through the segmentation process that I'm sure everybody loves here, where you have to define the anatomical regions based off of the grayscale values. From there, you generate your models. You can do some analysis and reporting within the software, do some scripting, automate it. We have some AI tools off the shelf to automate it as well. And then you export it out here. Our three main industries we work in, or main applications within the life sciences, are divided into three sections: implants, guides, and surgical planning. So these are more patient-specific guides, patient-specific implants that are 3D printed and used to treat patients during typically joint arthroplasty and things like that. Then there's point of care 3D printing. So that's 3D printing at the point of care within hospitals, and then in silico clinical trials. So this is where customers leverage simulation technology, finite element simulation technology, to create digital human twins and supplement clinical trial data to reduce cost of clinical trials, reduce the number of patients that are required for clinical trials, and just basically get products to market faster and more efficiently. So our primary flagship tool is Simpleware Scan IP Medical. So this is our FDA cleared CE mark tool, where you can import the image data, go through your whole process, and export your models. So this is the main product that most of our customers use within these areas. Let's see here. Okay, and this tool is cleared for clinical use with different printers and different materials and different anatomies. You can see here Formlabs, HP, Rise, Stratasys, all the different materials, and typically they fall within orthopedics, maxillofacial, or cardiac, our current approval areas that we have from the FDA. So what does the software do? Again, from kind of the left of this half circle to the right is where you would import your image data. You go through your segmentation process. You can do your different measurements and analysis. You can integrate CAD geometries into the image data, generate your models, whether STL, 3MF, NURBS, or finite element mesh, and then you can export out the STL or 3MF for 3D printing. And then within the tool, we have different types of automation. And that's where we start getting into how we leverage AI to scale up a lot of these workflows. Because traditionally, people, when they start going into an image-based workflow, regardless of the application, you spend time establishing that workflow and trying to make it as quick as possible. And then once people reach that point, traditionally the limit was, if you could write some Python script and script some of the processes, that's kind of where the automation ended. But now with AI tools and machine learning, we can take that to the next level and really turn some of these workflows into single-click solutions, where the segmentation is done, the landmarking is done. With the landmarking comes measurement and device placement, and we even are exploring using AI tools to automate the placement of devices within the anatomies to optimize that as well. So within the software tool itself, we have a 3D printing toolkit. So this is for people who want to do the anatomical 3D printing. So we're pretty proud of this. It brings everything you need for anatomical 3D printing into one solution. You don't have to export it out to add support structures or do Booleans or things like that. It's all within the software. It's all included. You don't have to buy something else. So everything is within it to help you save time with a nice ease of use tool contained within a single ribbon, and gives you traceability all the way from the image to your final exported model. So then we get into the automation. So the first level, as I mentioned, is scripting. So we have full APIs for Python and C# scripting. You can record a macro. You can convert from a log file that logs every single click you do. You can convert those into scripts. You can build plugins. We even do fully customized solutions for some of our customers to automate some of their workflows. Then we get into some of to what we call our second level of automation. So these are our off-the-shelf AI-enabled tools for automatic segmentation and landmarking. So these are tools where we procured data sets from some collaborators, and we created our own ground truth. So essentially, we segmented out the data sets, did the landmarking, got them reviewed by clinical experts to make sure our ground truth was correct. And then we trained some AI tools and now offer it off the shelf. So we have AS Ortho CMF. So this is automatic segmentation and landmarking of the ankle, knee, hip, shoulder, and CMF from CT, and then the knee from MRI. And then we have AS Cardio. So this is automatic segmentation and landmarking for 3D and 4D heart CT. And so these are turnkey add-on modules for Scan IP Medical, and it can really speed up your workflow. Again, this is trained off of our ground truth data sets. So depending on your data, the algorithm will perform quite well. It again depends on your data. You know, we've had some. So for example, for the heart, we've had people run it on. You know, it was trained on adult, relatively healthy hearts. We had somebody, one of our customers, which I'll show later, run it on a 16-year-old, and it worked perfectly. Then they did it on a 2-year-old, where that one required a bit more cleanup. But again, these are off-the-shelf tools in the software. It's basically like having a threshold button. You know, you run it and then you clean it up. Same with this one. You'll run it, and some cases it'll be minimal cleanup. Some cases it'll be quite a bit of cleanup. But the idea here is to eliminate the need to start segmenting from scratch, at least for these anatomies. And as we grow and build it, and we'll have a new, we have a 9-month release cycle, and we'll be having our next release in December, and we'll be having more anatomies added to this tool. So stay tuned for that. Then we come to our third level of automation. So this is where we offer complete solutions to our customers to automate their workflows. So we can automate the segmentation and landmarking of any anatomy from any imaging modality. So it's integrated into a tool we call Custom Modeler, which is a Simpleware software add-on module. So it's the base software. We train and build a specific tool based off of the requirements of the customer, and then add it to Custom Modeler, and then the customer is off and running. So it's built on top of our software infrastructure. It's not just a custom code that you with an executable or anything like that. So it's fully supported. And how it works is the ground truth is either provided by the customer, or we can procure it as well for their specific needs. So say somebody wants to do a shoulder. You know, they have a solution for patient-specific shoulder surgical planning. So the customer, they you know, typically have a good sense of what kind of data they see, what kind of pathology, things like that. So we get a ground truth data set from them. Either they segment it, we segment it, we get it approved by, reviewed by a clinician, and then we can train a tool to automate the segmentation. We can automate the landmarking, and also pull in any automation from Simpleware in general into that workflow. So we can fully automate their workflow. So we have customers where it just, it monitors a folder, which is within their network. When the users drop in the DICOM files, it pulls it in, processes it, and spits out the results on the other end. So some current examples, Corin is one that we can talk about. The OPS system, the optimized positioning system, they process 500, actually, I think it's more like 600 hip, knee, ankle CT scans per month. And we were able to reduce their time by 94% using the AI tool. So they've been customers of ours. It's going on 11, 12 years now. And they optimized it based on scripting. But then once we were able to bring in Custom Modeler, we really reduced the time. And then we have an orthopedic device OEM. Obviously, I can't share who they are due to confidentiality, but they processed about 5,000 cases in the first 3 months. So about 60 cases a day. So this is just when it was initially deployed. So we took them from about 30 minutes to below 5 minutes average per case. And then we're also exploring leveraging AI tools for device placement and device positioning to aid in that process as well. So it's not just the clinician going into the 3D environment and kind of eyeballing it. So at least this gives them a very solid starting point, and then the clinician can adjust as they see necessary. So these are the types of solutions that we're deploying within customers. So getting into some examples where, where the rubber hits the road. One is anatomical 3D printing in the hospital. So this is Nicholas Children's Hospital. Some of you might have seen this. We've been working with them for a while, Dr. Hannon and his group. So this is a case here where they're using our AS Cardio tool, our off-the-shelf cardiac segmentation tool, and our 3D printing toolkit. So they can automate the segmentation and then create their supports, create the base, do their text embossing, all within one environment. No need to jump around at all. And their manual workflow in this case here took about 2 hours. With us, with Simpleware, it took about 15 minutes. So significant time savings. Just a little quote from Thomas Hagelin, their engineer. Our new engineer segmented it. Well, he hit the go button on AS Cardio and then added the base and supports. Very quick. So these are nice examples of where these tools can be leveraged to speed up your workflows. But again, within point of care, we don't see quite the scale. We don't see people processing 5,000 cases in 3 months or 5,000 models in 3 months. I'm sure, I think Ben from Stratasys is on the call. I'm sure he'd love to see that, where people are going to that scale. But currently within point of care, it's still, bit more of a case-by-case basis. So there isn't quite the scale as, say surgical planning or patient-specific guides and devices. Then we get into implants, guides, and surgical planning. So scaling up to save thousands of patients, essentially. So the optimized positioning system at Corin, as I mentioned before, actually where they process about 600 patients per month. Again, you get the patient imaging. They send that to Corin, and then their OPS team processes it, and then designs surgical guides, a surgical plan, and then delivers that to the surgeon with the device for them to go into the OR. And I think they've surpassed, I think it was 30,000 patients last year. So I'm sure they're well beyond that now that they've processed. So in 2020, we deployed custom modeler to them, to Corin, and it was about 100 ground truth data sets were used for this. We trained the AI machine learning model, and we reduced time by about 80%. And then we did a second version because over time, as they, we kind of enable them to scale up, and then they can accept more patients and more variability. But some of those patients kind of fall outside of what we trained the AI tool on. So we retrained it with more data. And in this case, we did that, and we're able to reduce time per case by 94%. And about a third of that time is compute time. So just the computer chugging away. So really the limitation is hardware, not necessarily how many engineers do you have or technicians you have clicking away doing the segmentation. Recently, we did a webinar with Entropology. So we did, just an automated guide design workflow on 50 tibias. So Entropology has some nice technology where they can automate their design process because they're not a traditional CAD platform. So it's really interesting to see how they can leverage that and bring that into the life sciences with some device-based design. So in this case, we had, it was 50 tibias, and we just segmented it with AS Ortho automatically, sent that to them, and then they did some work to automate their design process as well. So it was a nice little workflow we did with them. So you might have seen a recent announcement where we're really trying to bring this out to users, and the best way to do that is students. So we've leveraged, Synopsys has a Synopsys University bundle, which they offer, and we've been able to become a part of that with the Simpleware University bundle. So the goal is to inspire and foster the world's next generation of technologists and innovators in this space. So the problem that we're trying to address is that converting 3D images into models for academic research requires sophisticated software and well-trained students proficient in said software. So, obviously students, I was a grad student, students are cheap, throw more students at it. It's a learning process, bang your head against a computer. But, and one thing we did was we played musical chairs with the one computer. I used Simpleware when I was doing my PhD. Oh man, 14 years ago. And we played musical chairs at the one computer where we could afford to have a license installed. So complex licensing and high cost limits researchers' access to software and their ability to conduct research and train students. So we wanted to address that. So we consolidated all the Simpleware academic products into a single, very low-cost bundle to reduce the barriers of access. It's 50 full floating network academic licenses of Simpleware, no technical or size limitations, it's $1,500 per year. It includes AI, auto segmentation tools, AS Ortho and AS Cardio. So it really enables campus-wide access for enhanced collaboration and teaching. And currently it's available in the Americas, Europe, the Middle East, and Africa. So we do not have current plans for Asia Pacific, but hopefully we can work on that. But currently that's where it's currently available. So if this interests you, if you're an academic and you're spending an arm and a leg on software, contact us. And I think this is going to be a significant game changer. And I hope just enable more people to do research, because a big complaint I hear from professors over and over again is lack of transparency in pricing and variability in pricing, that some groups pay 10 times more than others for opaque reasons. So hopefully we're just going to standardize that and have this available for academics and students. I wish I had this when I was a student. So I think I'm over my time. So I'll just skip this here and I'll just thank you for your time.
Thank you, Karim, for the excellent presentation. By the way, for all the speakers and everybody else, feel free to enter your LinkedIn, social media link, whatever, into the chat box. That chat box is simulating the physical network and networking experience. I have I'm really excited about this education bundle or university bundle you were talking about, Karim.
Mm-hmm.
And I know we have a couple of students in the audience. You said 1,500. Is that just for one license?
It's for 50 licenses.
Oh wow, 1,500 for